So welcome back to part 2 of our lesson on fungi. Now we're going to talk about fungal ecology, and then we'll also get into specifics about different types of fungi. So most fungi, they are decomposers, very important in the ecology in order to release nutrients back into the life cycle. And then we'll look at some of those, and then we'll also look at the fungi that form symbiotic relationships. So remember, it's a relationship that they form with another organism where they're either going to get benefits from or not harm the other one. All right, so here we go. We'll look at mycorrhizae and we'll look at lichens. Okay, sorry about that. I had to pause for a minute. My dogs wanted to come in, see what was up. Okay, here we go. So decomposers. Without the fungi, the earth wouldn't be functioning as it does now. They're very essential components to the ecosystems, and they work with the bacteria. So when we looked at cycling of nutrients in the previous unit, we mainly focused more on the bacteria, but fungi are right there with them. They absorb nutrients from organic wastes and dead organisms, and they're really important in like our plant. When plants die, they can degrade the cellulose and the lignin. Remember, those were the main components in the plant cell walls. Okay, and when they do degrade these bodies down, they release the water, they release the carbon dioxide. So they're part of that hydrologic cycle, they're part of that carbon cycle, and they also release other mineral components of the organic compounds. So all of these things get recycled back. So therefore, these essential nutrients, they don't get locked up in the huge mounds of animal carcasses and feces and branches and logs and leaves. Right, so it's very important. So again, here you can see there's a dead log. You'll also often find fungus growing out, and those are the fruiting bodies that are growing out from the log. So that means embedded within the log is all the mycelium inside of there going to work, releasing their enzymes to break it down. Okay, and then you can see that on the orange occurring there as well. So again, some we said are form mutualistic symbiotic relationships. They're usually mutualistic. So one type of mutualistic relationship is called mycorrhizae. So these are fungi that are associated with the roots of plants. And so that's what you can see here is you can see the plant roots, and then scattered all throughout are the more fine hyphae that get associated and form a mat around the roots of plants. Down in the picture at the bottom, B, you can see on the left side plants without mycorrhizae, and they're very much reduced. And on the right side, I'm sorry, did I say left? I'm not sure if I said left side before of B. So that's on the left side. On the right side, you see plants that have mycorrhizae, and they're doing much better. They're growing much better. That's because there's all this decomposed organic material in the soil, and so the mycorrhizae have an increased surface area, and so when they absorb the nutrients from the soil, it can get passed on into the plant's roots. So it really then increases the surface area of the plant's roots itself. So the plant therefore can absorb more water and more nutrients. From the other end, because it's mutualistic, so both benefit. On the other end, then the roots are supplying the fungus with nutrients that they're making. So the nutrients that they make, the organic nutrients, can move into the fungi body, the fungal body. So very mutualistic situation going on there. Remember that we looked at nitrogen cycling. There was also phosphorus cycling, And so one of the ways that those nutrients get into plant roots is through the fungi mycorrhizae. You can actually, if you're growing plants, you can actually buy a starter mycorrhizae to put into your soil, and that oftentimes then helps to enhance the growth of the plants that you're trying to grow. So you can look for that in nurseries, ask about that. Okay. Another form of mutualism is through an organism we refer to as lichens. Lichens isn't actually one single organism. It's actually a symbiotic relationship with either green algae or cyanobacteria. And so green algae and cyanobacteria are single-celled organisms that are able to photosynthesize. Okay, and we're going to learn about those later in the semester as well. So these are, again, organisms, single-celled organisms that can photosynthesize. So the fungus kind of grows around the algae or the cyanobacteria get embedded in there. So the autotroph is photosynthesizing, making glucose out of carbon dioxide and water, and then that compound can be absorbed by the fungus. They believe that it's a mutualistic situation here. It's unclear exactly how the photoautotroph then benefits from the fungus. It has been suggested, though, that the fungus actually helps the autotrophs to obtain water and other nutrient minerals. And the fungus, therefore, is protecting these single-celled organisms from desiccation, both by surrounding it and then also by supplying it with water and the minerals that it needs. Okay, there are actually some researchers that don't believe that it's mutualism. They think it's a type of controlled parasitism of the autotroph by the fungus. So the jury's still out on that. So I just wanted to zoom in so again you can see a little bit more closely how the fungus and the algae are related to one another. So you can see there's a layer of fungus and the algae are embedded in there. But the algae, the sun does need to penetrate through so that the algal cell or the cyanobacterium can photosynthesize. There's some small pictures there. I found a nicer picture. So you might see lichens on trees, on rocks. So areas where you wouldn't really think algae could grow, and they can't grow there alone, or cyanobacteria. So on the bark of trees, you might see this type of structure on there, and that's actually the lichens. A lot of times people mistake it for moss. Remember, moss needs water, and so this on the side of a tree or on a rock or something, it may look dry to you, but again, it's the fungus that's able to retain the water in their hyphae, which will then could benefit the photoautotroph. Okay, so that's not moss on the side of the tree, you see, that's actually lichens that are growing on the side of the tree. And you can tell the difference. I mean, now you know the structure of mosses. You can look really closely and see the gametophytes. But one of the easiest ways, one of the things I do is if it's just a dry rock, that's got to be lichens. And as you get to learn lichens more, you get to learn their structure and tell the difference from mosses pretty easily. And there's different types of lichens that take on a little bit of a different shape. But they're pretty interesting. And also what's interesting about lichens is they don't tend to do well in polluted air. So when you see lichens around, that should tell you that the air is pretty clean. So you want to see lichens, you want to leave them there. They're pretty, kind of gives you like a fairyland feel. But it tells you that the air in that area must be pretty clean, so it's going to be healthy for you too. Okay. Some fungus are pathogenic. There's about 5,000 species that cause crop diseases. So mostly the way fungus affects us negatively is by attacking our crops. And the problem with them is, again, they release these spores that can just spread by wind or other means. So when there's a fungus on crops, it can quickly spread along the whole field pretty quickly. So it's something farmers to really try to control for. There are some human diseases regarding fungus. Those that affect our skin, we would refer to those as dermatophytes. Some of you might be familiar with athlete's foot. We call it athlete's foot because it's commonly picked up in locker rooms. So the fitness centers, at the gym, in the locker room, or if you are participate in a sport, everybody comes together in the locker room. And again, you need moisture. The organism needs moisture to survive. So what do? You take a shower, you walk around, you're dripping wet. So the carpet in there, the ground tends to be kind of wet. You can easily pick up the spore, you can see on the bottom of the foot there, and get athlete's foot. We have creams that can kill that and take care of that pretty quickly. That's a pretty bad case right there. Ringworm, you see the one in the arm above. That common name is kind of a misnomer because you think ringworm and you think you're infected by some type of worm. It's not. It's actually a fungus, but it grows out in these successive rings. So even if another animal like your cat gets ringworm, you have to be careful when you're treating your cat because you could pass that to your own skin. So you have to be very careful about it. There are a few that cause diseases inside of our bodies. So there is a type of pneumonia, usually develops with people with a compromised immune system, such as AIDS. That is a fungus that can cause that pneumonia. That gets into the lungs. Pneumonia is really just fluids building up in the lungs. So it's the fungus that can do that. On the canvas at the end of this PowerPoint and below some of the videos that I posted, actually no, at the end of this PowerPoint, there's a link, or you can listen to it right from Canvas, to a podcast that's really interesting about how mammals may have evolved after the dinosaurs died out, and why mammals then had an advantage over the smaller dinosaurs that didn't die out. So they were actually after we call it the extinction of dinosaurs. They actually didn't all go extinct. There were some small ones that survived along with the small mammals. But the mammals then adapted for some reason, and there's a hypothesis that it might have been because of fungus. So I encourage you to listen to it. It's really, really interesting. And then we'll talk a little bit more about evolution too at the end of the semester. We can bring that up. But go ahead and listen to it, and I'll talk to you about it in class. All right, so let's look at specific types of fungus. And first one that we'll look at happens to be a pathogen. It's called a chytrid fungus, also known as Chytridiomycetes. A mycete refers to fungus. Okay, so most of these are unicellular, so that's what you see in the picture on the right side. Those are several unicellular chytrids. Some actually then can be composed of a few cells that end up forming a thallus. Most of them are, so their body is a thallus. We're not going to worry too much about that. Most of them are decomposers, and they degrade the organic material in ponds and damp soils. But there's one in particular that is parasitic to frogs, and we may be witnessing the extinction of our amphibians, mainly the frogs. We've been seeing several species around the world go extinct because of this chytrid fungus. When they're tadpoles, the fungus seems to get into their skin, and then as they grow, it compromises the skin, and so then the frogs die out. It's a pretty serious situation, and researchers are desperately, on the one hand, trying to learn more about the chytrid fungus and how it spreads, and other researchers are actually trying to capture some of these few wild frogs that are left of certain species and bring them into the lab to protect them and try to increase their numbers, try to reproduce them, have them reproduce in the labs so that we can save the species before a chytrid makes them go extinct. If you want to know more about that, I can provide more information on the Canvas site. Sad but interesting as well, especially I know some of you said you wanted to go into herpetology. This is a pretty interesting situation going on with frogs now. Hopefully we'll get the better of the chytrid fungus as time goes on. Okay, the next type of fungus we want to look at, they're called Zygomycota. Zygomycota. Okay, so here are the characteristics of the Zygomycota. Their hyphae tend to be aseptate. Again, that means like one long tubular hyphae with no divisions in there, no septae. We refer to them as coenocytes. Okay, so that's characteristics of the Zygomycota. They tend to produce asexual spores in sporangia. We're going to refer to those sporangia as zygosporangia. Okay, and then again, they produce spores, and we refer to those spores as zygospores. Okay, the zygospore then is usually produced sexually. It undergoes meiosis, and it's going to produce the haploid spores. So most are saprobes in the soil, again, decomposers. And there are a few that are actually parasites, parasitic. Okay. They also include molds that grow on food. Okay. And so here you can see this is like a close-up of black bread mold. I think a lot of us are familiar with that, with this mold that grows on our bread. So I'm going to explain all the structures that you're seeing here on the bread. Some of them are dangerous pathogens, so you don't want black mold developing in your house because, again, it's the spores that can be dangerous to you. So let's look at a close-up of this black bread mold. This is what it would look like. So basically what happens, you can see spores being released at the top there, and when it lands on a surface like bread, bread has good nutrients inside of it. It's moist, so it's a great surface for this mold to grow on. So when it does, the spore develops hyphae, and that then begins to branch and branch, forming that mat of mold called the mycelium. Most of the mold is penetrated into the bread, but again, you can also see some along the top. If I go back here, more of the white filamentous material that you see there, that's the mycelium. But again, most of the job that it's doing is penetrated into the bread. So again, they're releasing enzymes to break down the nutrients outside of the body so that they can then absorb the nutrients into their body. Okay, when they're ready to reproduce, some of those hyphae will divide and grow upward, so that becomes the aerial sporangia. And so the sporangiophore is the stalk, and then the sporangium is that black portion at the tip growing the spores inside of it. So it's dividing and dividing to create those spores. And there you can see the microscope slide, a close-up of the spore of the sporangium, and you can see all the little tiny dots there. Those are the spores itself. So this is why we call it black a bread mold, because the sporangium is black, and so those are all the little black dots that you see at the surface of this piece of bread with the mold on it. Okay, and then again, the spores themselves will be microscopic, so you may not be able to see the spores getting released. At any one time, there are billions of spores in the air, and you're constantly breathing spores in and out. So it's amazing with how many spores there are. Again, most of them are not pathogenic to us, but some of them are, and those that are can be pretty dangerous. Okay, so let's look at a little more closely of zygomycete and how it reproduces. So again, it can reproduce both sexually and asexually. So the asexual reproduction is easier. Again, what I had already mentioned, when the hyphae produce sporangia up into the air, then they produce the sporangia at the end of the sporangiophore, and within there they're just dividing the cells by mitosis to make the haploid spores. Remember that the cells of fungus are already haploid, so they're just dividing in order to make the spores, and it fills the sporangium. Okay, then those sporangium will break open and the spores will just be dispersed into the air. Okay, and so wind currents can take them away into the air. And when they land on a suitable place, like in this case bread, they germinate into hyphae. So you may have spores on the surface of your counter and you don't even see them. And if you haven't wiped it up in a little while, the spores will be on there, and that's not a suitable surface, but the spores are able to remain alive because the spore itself is protecting the chromosomes inside of it. So then you put down a piece of bread, right, and you leave the bread there, the wind can blow it or it picks up the spores right from the counter, and that's how your bread will get moldy over time. Right? So again, the hyphae get into the spores get into the bread, they divide by mitosis to form the hyphae, and it just produces more hyphae and produces that mycelium infusing through the bread, and the cycle starts all over again. Okay, so again, when it produces asexually, it's not changing the DNA at all. So they can produce very quickly and spread out and produce more structures, but the new cells are the same as the parent plant, so no genetic changes going on there. But here you can see that they can also reproduce sexually. Okay, so if you look again, so this is for our zygomycetes, a life cycle, maybe something you should take notes on. And you can see number 1 in the upper left corner, where the spores are germinating to produce the haploid mycelia. Okay, so we're in a haploid stage now. So remember then that the hyphae of 2 different organisms that are compatible with one another, and there's where you can see the plus and the minus. Those 2 different mating types will grow toward one another, and they attract one another because they can release pheromones. That's how the mycelia know if it's a compatible mating type. So they're attracted and they grow toward the pheromones or the odor, the chemical that gets released. I shouldn't say odor, the chemical that gets released that the other one can detect. Okay? So again, they grow toward one another, and then it forms a structure called gametangia. Gametangia. This is where the zygospore is going to develop. So it starts as gametangia. So when they meet, they form that structure, and then they can fuse through that plasmogamy. So the gametangia from both sides fuse together, okay, beginning to form a zygosporangium. Okay, so remember that each of the hyphae from the positive and the negative side, they each have a haploid nucleus. So when they form the gametangia, those 2 haploid nucleus are coming closer together. The plasmogamy is when the structures fuse together, the cells fuse together, but the haploids stay distinct. The haploid nuclei stay distinct. So now you see we've moved into that yellow, or we're moving into the N plus N, but we quickly then go through karyogamy. So our nuclei are forming, and now we have a diploid organism. Okay, we're in a diploid organism. And the zygosporangium keeps developing, so it gets this big black structure is what it looks like. And that's got maturing zygospores inside of there. So it's 2N. So the zygospores now within there, they are dividing by meiosis. That's why we've moved up into the haploid again, into the purple zone. Meiosis is occurring, so all of those spores are developing inside of there, and it breaks open, the zygospore breaks open, and it releases those spores, and it starts all over again. Okay, so the zygospores that develop, that big black structure. It can be dormant for several months. So again, another structure that can survive desiccation, extreme temperatures. It's believed that the meiosis is probably occurring just before germination of the zygospore. So they divide very quickly just before they germinate. And then after germination, the aerial hyphae develops within a zygosporangium at the tip. Okay, that's on number 7 there. Kind of skipped that before. And again, it uses mitosis to produce many haploid spores. So it has to use mitosis because we're already in that haploid section. Okay, so really kind of follow along what stage we are. You can go by the colors here. In the purple, that's haploid, and in the yellow, that's diploid. So if you get confused about which type of reproduction occurs or what type of cell division, haploid or, meiosis or mitosis, well, if it's haploid and we're dividing cells, remember, we want to maintain at least the haploid. You can't go less than that because then it's not enough chromosomes. So that will divide by mitosis. But in a diploid cell, if we want to produce haploid cells, then we divide by meiosis. Okay? The only time when diploid cells divide by mitosis is when they want to create more diploid cells. Okay, so think about it, just the math, whether you're dividing something in half or not. Okay. So again, back to that black bread mold, it's the zygospore and the zygote itself that are diploid. All the hyphae and the asexual spores are haploid. Okay, so you may have to go through this cycle a few times. Again, you really have to understand the terms really well before you can remember the cycles. All right, here's another type. This is a type of fungus called Glomeromycota. This is an interesting one. It's what we might refer to as arbuscular mycorrhizal fungi, and sometimes it's just referred to as AM fungi because it's a tongue twister to say arbuscular mycorrhizal. What that is, arbuscules are structures that help plants capture phosphorus, sulfur, nitrogen, micronutrients from the soil. So this goes back to the mycorrhizae we were talking about. So this is the specific type of fungus that can form mycorrhizae. So again, here now you can see is that under where it says ability of early plants to live on land may have depended on help from the fungal associations. So the larger cells that you can see there, that's from the roots of a plant. So you can see they're very thick. And then even when a plant extends out a root hair, it's relatively thick. The benefit of the mycorrhizae fungi is their hyphae are much smaller, much thinner. So they can penetrate smaller spaces through the soil, have much larger surface area, and therefore an easier time absorbing the nitrogen, the phosphorus, even sulfur, other nutrients. And then as it comes through the body into the root can benefit from some of those nutrients. Okay. So it's a pretty neat situation to help plants get nutrients. And then again, the roots store the nutrients that the plant makes itself. It makes carbohydrates and things. And so the fungus needs that also as a nutrient. So it benefits by absorbing some of the nutrients from the plant. So mutually beneficial in this case. Okay, the next group is called Ascomycota. Okay, Ascomycota are named for their specialized type of sporangia. They're called asci. Okay, there are these sacs, and you can see in the arrow from the cup-like structure, that's the fruiting body. And so if you take a microscope and you look below, you can see that fruiting body is made by several of these little sacs called asci that are smashed together, but within the sac are the ascospores. Those are the haploid spores inside of the ascus. Okay, So those are sexual spores inside of the ascus. So we refer to those as ascospores. Okay. But also single-celled yeasts are also considered Ascomycota, and that's the picture you see in the lower right corner labeled C there. So those are single-celled yeasts, also Ascomycota. So they stay single cell. They don't form these multicellular fruiting bodies that you see. So they remain very simple. Okay. Little more detail on what that ascocarp looks like. Okay, so the ascocarp is the fruiting body, is that structure. And again, you can see it's circled, and we can look at the microscope slide. So that whole structure is made from the hyphae coming from the mycelium below, and then they differentiate into the sac. That's an ascus, and within the sac are ascospores. Okay, all those little circles that you see inside of there, the haploid ascospores. And here's just another close-up of that structure that you can see. The spore inside of a single ascus, so there are many asci there. Okay, so again, you got to get your spelling down. If I'm talking about several of those sacs, plural is asci, A-S-C-I. If I'm referring to one of them, it's an ascus. Okay, so it's important to understand the structure before we get into the life cycle. Okay, let me show you another structure that can be produced. Penicillium is also a type of ascomycete. And so here we're going to see conidia that are formed. Okay, and so this is more the asexual form of reproduction. So the hyphae will branch and form the aerial conidiophores. And then at the end of the conidiophores, they will divide and produce the individual conidia. So those are spores. Okay, so if you remember, the general term we use for a structure that makes a spore is called a sporangiophore, sporangiophore. So conidiophore is a type of sporangium. Okay, and so then sporangia form spores, so a conidia is a type of spore. Okay, so we use the asexual reproducing portion of a Ascomycota uses conidiophores and produce conidia. Alrighty. All right, so here's the asexual reproduction. Again, very simple. The hyphae will then branch and form the stalk, which is the conidiophore. And at the ends, again, is where they will divide by mitosis because they're already haploid and form the conidiophores, form the conidia, which are the spores. So the spores then get released into the air and get carried away, where they can then, when they hit a suitable surface, they can then form new hyphae. And again, they're going to be genetically identical to the parent, and they remain haploid. Okay. Here's the sexual reproduction, the life cycle of a ascomycete. Okay, and so it's similar to what we saw with the zygomycetes. We're not going to see zygosporangia, obviously. Again, those things are named for the type of organism that they are. So you see again, number one over on the left side, two compatible hyphae from different mycelium bodies, different individuals, will grow toward each other. And again, they're compatible, they're releasing pheromones, so that's how the fungus knows that it's compatible. So they'll grow toward one another. The tips will fuse by plasmogamy, okay? And that combines the nuclei of the two genetic types. There'll be the haploid nuclei will be inside of there. Okay, so when you remember earlier, the earlier video, we said when two haploid nuclei occur inside of one cell, we call that dikaryotic. And so the mycelium now can produce, this can divide into mycelium and produce dikaryotic mycelium just by mitotic division of both types of nuclei. So you're just forming more and more of the nuclei, and the hyphae are branching out. Okay, so then look at number 3. You'll see some of the dikaryotic mycelium then will start to produce the fruiting body known as the Ascocarp. Okay, so the Ascocarp begins to develop, forming that mostly what we see is that cup-like structure. Okay, and within that structure, the hyphae now are producing those little sacs that are called asci, and they contain the haploid nuclei of that they got from that merging of the two hyphae. Okay, and then within the sac, the haploid nuclei fuse. So now that's diploid. So it was dikaryotic because it was N plus N. The two nuclei haven't fused yet. Once they do fuse, then it's a diploid zygote. And you can see the arrow there is bluish. It's a very short segment. So the organism is actually diploid for a very short time, because it very quickly then goes into meiosis. Meiosis again will then divide the cells or divide the nuclei. The nuclei will go through meiosis, not the cells themselves. So the nuclei will go through meiosis, and that is going to create the haploid nuclei again. Okay, so what's the point of that? We went from haploid to dikaryotic to diploid right back to haploid. Again, it's shuffling the genes, so you're getting a little bit of different genetic variation every time it goes through that. Okay, so again, you've got the 4 daughter nuclei now that are haploid. Now those can continue to divide by mitosis just to produce more of them. That's why in that sac you can get so many of them. Okay, then they'll get released, they'll get dispersed out. Sometimes water hits it and pops it open, and then it gets dispersed into the air, and then the cycle will start all over again. So again, in the figures such as this, what they're trying to do is use color so you can keep track of when it's haploid, when it's diploid, and remember when it's dikaryotic, we call that heterokaryotic. It means different nuclei in there, and they haven't fused yet. It's different, but we know it's 2, so you can just call it dikaryotic. Okay, so here's a little more close-up on the Astrocarp. Again, the Astrocarp is made out of the hyphae. The ends of the hyphae within the Astrocarp will differentiate to form that sac. The sac is called the ascus. That's where the diploid, first the dikaryotic nuclei will fuse to form a diploid zygote, and then through mitosis, the nucleus divides, forming more and more haploid nuclei. The nucleus divides by meiosis to form several haploid nuclei. That's why you can see right inside of there, it shows 8 ascospores inside of that ascus. And again, there's several of those asci that are forming the fruiting body, which is called an ascocarp. Okay, so again, it's the vocabulary that's going to hit you. So you gotta get your vocabulary straight. Okay, this is the last group that we're going to talk about. We think this is the most recently evolved group of fungi. They're called Basidiomycota. These are the ones you're probably the most familiar with because they produce the mushrooms. The fruiting body are the mushrooms that we are all pretty familiar with. Basidiomycota are very important decomposers. There also can be mycorrhizae partners. So it's not just the other group, but Basidiomycotes can also be form mycorrhizaes. But usually when you're walking along a damp lawn or something, you see that mushroom pop up. So that's a Basidiomycota organism. Okay, and so here you can see a puffball and you can see a stinkhorn over to the right. So they're very diverse. in their fruiting bodies, and they can look pretty cool. As you walk around, just start to notice if you see a tree limb that's fallen, or a trunk of a tree, you'll see fungus growing out of it because it's breaking that down. Those of you who have eucalyptus trees in your area, sometimes you'll see yellow fungus, shelf fungus growing out. It looks like a shelf. That's why it's called shelf fungus. That's not good in a living tree. So it's eating away the inside of the eucalyptus, and that can cause the eucalyptus to weaken over time and fall down. Well, our eucalyptus are very tall. They can be 60 foot tall. So if you see fungus, you want to get to the nursery, ask them for some kind of fungicide so that you could kill that, so you don't want that eucalyptus tree to fall down. Okay, so again, terms. So the structure that grows the spores, okay, are called basidia, and then the spores themselves are called basidiospores. Okay, the mushroom is called a Basidiocarp. The mushroom itself is the fruiting body, and more appropriately, it's called a Basidiocarp. Okay, there are connections that help distribute the nuclei during cell division. This is a little distinction between the other groups. You don't have to really worry about that, clamp connections. But they do reproduce asexually, and there's various types of spores. Okay, so we're going to look at just a few. We'll look at one representative group. Okay, so again, the diversity is very important here, and also these are the types that we tend to eat. So here's a little more on the Basidiomycota, on the Basidiocarp. Right, so that's a mushroom, and when you look at the underside of a mushroom, when the bottom kind of opens up, the stem is pointing straight out you right now. There are gills on the other underside. So it's along those gills. If we look at the microscopic slide, that's one little gill flap there. You can see that that's where the basidium are located, and then the basidiospores are growing off of that. So some of those basidiospores are already dislodging. Okay, so that's why it's the fruiting body, the part that's reproducing in this organism. So the body of the Basidiomycota is the mycelium, and that's under the soil. You don't really see that. You don't know the fungus is there until the fruiting body develops. And that fruiting body then, like I said, can come up from the soil. Here I'm showing you the shelf fungus to the right, growing out of the tree. I mean, they can be beautiful. These colors are real. Here's some fungus growing out of corn. So again, it can damage our crops pretty easily. So that's the fruiting body coming out of the corn. This is an interesting one. This one's called an earth star. It's pretty cool. I put a video on Canvas so you can see. I mean, it's about the size of your hand, and it starts out closed like a fist. And then when it's ready to release its spores, each of those little fingers will open up like that. And so I have a video where you can see that happening. They're really, really interesting. So again, I can't put enough video or enough pictures to show you the diversity of the Basidiomycetes or Basidiomycota. So gotta look at the life cycle, right? Put the life cycle in there. So in this case, what's dominant part of the life cycle is the dikaryotic mycelium. So the mycelium itself is dikaryotic. That's the main portion of the plant. And you can see that down toward the right side of the circle, you see the little A where it says cytoplasmic fusion. Cytoplasmic fusion. So once again, the hyphae of compatible positive and negative hyphae grow together. They fuse, the cytoplasm fuses by plasmogamy, right? And that creates new cells for the mycelium that are N plus N because they start out haploid. And so once they fuse, the two haploid cells now are within the cell, and we refer to them as N plus N. Okay, this then starts the producing the structure of the fruiting body, okay, the mushroom or the Basidiocarp. Okay, so that grows by mitosis and extends up through the soil, producing the Basidiocarp at the surface of the soil. Okay, so it's within there that underneath in the gills that these cells begin to form. They're kind of club shape, and they each hold a dikaryotic form in there. They each have the N plus N, and that again, that grows at the margin of the gills. So that's what I was showing you. Whoop. I gotta go back a couple. That's what I was showing you here, the gills underneath there. So again, this is the close-up. So along the edges of the gills, you can see the basidium growing, those little greenish bumps. And then that's where the basidiospores will begin to develop. All right. Okay, so that's where we are at C up on the left side. So along the margins of the gills, the basidiospores are developing. Okay. As they're developing, there's a portion where the dikaryotic cells merge, forming a diploid stage, but then those divide by meiosis. So again, we're going to go right back to haploid, and that's where we're creating more and more of the spores. Okay, and that's how we can create a lot of spores. Alrighty, and now you can see over on the right side those little basidiospores developing there. That's what's going to get released, and then there's usually a gust of wind or something as the mushroom cap opens up more and more. A gust of wind comes through those gills, carrying the spores away. And again, I put another video where you can see that happening. Okay, so again, you want to just keep straight. When is it haploid? When is it diploid? When does meiosis occur? When does mitosis occur? Okay, so it's very similar to the other life cycles that we've seen, but we just have different structures. So in the Basidiomycota, the fruiting body is the mushroom. In the Ascomycota, the fruiting body is the Ascocarp. Okay, and they form asci. Alrighty, in the Zygomycota, the fruiting body is a zygosporangia, so they produce the zygosporangia. So you want to just kind of keep those things straight as you're studying your fungi. Okay, that's it. That's all I've got for you. And have fun studying fungus. Previous students always tell me they remember the fungi the most, and I think it's because it's what you're least familiar with, and so you end up learning the most about it. All right, everybody, have a good day.